Okay, everyone. Well, let's go ahead and get started here. So it's 12 o'clock. So we're back at it here in biochemistry. So let's start with a couple of announcements to begin with. So first of all, this is going back a week actually. Last Friday, I posted an announcement. Actually, I probably have already mentioned this to you all because I forgot that you would have been here. So anyway, but the university has decided when we're gonna get back together face to face. Okay, and on Canvas last Friday, I followed up their announcement with an announcement on Canvas and what that would look like for this class. And what it essentially means is for this section, section 008, we will meet for the first time face to face, not next Friday, but the following Friday. The reason that next week is, of course, exam week. Okay, now that's another announcement. So next week is exam week. I've posted two announcements on Canvas about that, one yesterday and one today. So make sure you look at those announcements and read through those. The first one that I made yesterday gives you the structure of the exam, tells you how it's going to be proctored, the information you need to get prepared for the exam, to have it proctored by Respond as Lockdown Browser. I give an extra credit little assignment for making sure that you if you do that extra credit assignment, you'll get 2 points extra credit as long as you do it before 11:59 p.m. So make sure you look at that announcement and do that assignment. You might as well. It's worth 2 points to you. Okay. The second thing in regards to the second announcement, I'm going to run a Q&A session next week, a special one. I'm going to run it on Tuesday at 12. We'll do it in this environment, a Zoom environment, because again, I can't bring everyone together in a face-to-face environment. And I will take attendance at that for extra credit. Okay, it'll count towards the extra credit that we have accumulating all semester long. And we'll hopefully you'll bring questions and I'll be able to go through those questions, as many as I can in that 50-minute period. Okay, so that's going to be at 12 o'clock on Tuesday, the day before the exam. Okay, so that's it for announcements. Let's go ahead and look here at our riddle for the day. So I'm taken from a mine and I'm shut up in a wooden case from which I am never released, and yet I am used by almost everybody. What am I? And I think I have someone in the chat. Maybe they've said that. Okay. So, oh, yes. So this would be for the Q&A. Can we join late if we have a class till 12:15? Yes. So I'm still looking for an answer to the riddle, though. What about an answer to this riddle? The other sections have got it, so don't let me down. Section 008. Lithium, close. It's close to that. All of you will probably use it today. Lead, there it is. So lead is right. And you think to yourself, why a wooden case? Because pencils have lead. So, and some of you are saying, no, pencils don't have lead anymore. They don't put lead in pencil. This graphite. I know. Okay, so, but anyway, that's what I was looking for. Okay, so today, some awesome stuff to do today in biochemistry with our worksheet. And of course, I think that because I am the geek, remember, okay, but we do have some stuff that I really think is pretty cool to go through today. So what we're going to be essentially doing is we're going to be looking at this peptide, what you see here, a very small protein, only 9 amino acids long. And we're going to essentially try and map that onto an alpha helix. Okay, now broader principles as we start to do this, okay, to think about. Within this class, my hope is that every student would walk away from BC351 with some understanding of the structural characteristics of proteins. Okay, so that's certainly some of what we're going to be thinking about today. We'll be specifically thinking about an alpha helix. Okay, but that will be more broadly applied to lots of structures that we'd find in proteins. The second thing that we're thinking about, and that I'd hope people would take away from a class like this, is that there has to be interactions that stabilize protein structures that allow them to form. And so we're going to be thinking about some of those interactions today. And I would want all of us to have some grasp of the interactions that are found within proteins that are stabilizing them. And so we'll certainly do some of that today. Okay, now the main question is this. if we were to map this peptide sequence, okay, that you're looking at here, and that's also put in your worksheet, if we were to put that onto an alpha helix, would the alpha helix form? Okay, and that's a question that you are going to be doing here in just a moment when I send you into your breakout rooms. Okay, now some helpful information here, right? So just in case, for the single letter abbreviations, I've given the 3-letter abbreviations, right? And I've given PKRs also of amino acids within this peptide that are either basic or acidic, okay? PKRs, by the way, I will be giving, and PK1s and PK2s, I will be giving those on the exam, just so you know, okay? All right, so let me go ahead and open up our breakout rooms. I'm going to go ahead and give you all 10 minutes to work on this, all right, and answer that question. Can this and will this adopt an alpha helix? Okay, so rooms are going to open right now. Okay, so go ahead and jump in a room if you don't have that option. Wait for me, and I will start assigning those as I see people kind of filtering down in those rooms. Okay, all right, 10 minutes. Will it adopt the helix, and then answer some of the other questions on it as well. Okay. I'm going to go ahead and start assigning people to rooms as I see people kind of hanging out in here. Some of my rooms got really big. Hey, Grant.
How's it going?
Okay, how about you?
Doing well. We're getting over all my midterms. I have one more exam. I took 3 this week. So.
3, wow, busy.
Yeah, pretty miserable. You missed it.
You got one more today?
Yeah, one more at 1. It's physics. It's not too difficult. I'm not too worried about it.
Okay. All right. I have students in my other class that said they got that physics exam, so I was wondering if you did too. So he said that you had one today. But room 2 got big. It's like 10 people in there.
Wow. I wonder how that happened.
I don't know. Should I start? Well, they just all jumped in. Who knows? We'll just let it ride. Well, good luck on your physics exam today. Is it physics 122? Is that what you're in right now?
1.2, yeah. But professor is probably the most lenient physics professor you can get. So.
Is it Brian Jones?
Oh yeah. I really like him. He's a great guy. I like the way he talks too. But I like the whole holistic grading. It takes a lot of pressure off the exams.
Yeah, I've heard really good things. I mean, I know Brian, so yeah, he's done really good things in physics for sure. So very popular.
Yeah, I actually found out that my roommate's younger brother is in this Zoom call because he kept sending him my old roommate videos of me through Zoom of me like doing the TA thing, and I thought that was really funny.
Oh, he's so
yeah. So yeah, it's pretty funny. Yeah, I was like, oh, I didn't know that your little brother was here. It's like, yeah, he keeps sending me videos of you.
Huh, that's interesting. The small world we live in, sure. Okay, well, I guess we can start to jump into some of these rooms here and see if they have questions. I'll go ahead and start at the top.
Let's go. It's number 13.
Okay, sounds good. See you in a bit.
kids now.
This test is gonna be so fun.
I know. Oh, hello.
Oh, any questions that I can help in here with?
We're just kind of looking at like the charges and everything of all the amino acids
Okay.
right now, so we can figure that out.
Okay. Remember, in this, because it's a peptide, you only need to consider side chains.
Okay.
Because in the peptide, you have the alpha amino group and the alpha carboxyl group will form a peptide bond with each other, and so you lose PK1 and PK2 of those. Okay.
Okay. Yeah.
Got it.
Awesome. All right. Well, I'll leave you to it. And if you need anything, let me know. Okay.
Perfect. Thank you.
Yep.
L and A were, what were they?
Those are also non-polar, right? I think.
So the amino acid A is nonpolar, L is nonpolar, and I is nonpolar. R is basic, and E is acidic.
Okay.
So then I went through the way that they're ordered. So A would pair up with I, R and E, L and A.
Hello. Hi.
Any questions that I can help out with in here?
So kind of just like what I was thinking was, I think it was like 2 or 3 lectures ago, we talked about how that one alpha helix couldn't form because they were all positive and it would break the hydrogen bonds. So I was kind of thinking along those lines, how there's only one positive one, I think, and the rest are either polar or non-polar. And then we were looking at the delta delta G's, like that chart. So we were thinking that it could make an alpha helix.
Okay. Yeah, you're headed in the right direction. You definitely want to start to think about these side chains, right? And what kind of
Right.
side chain interactions they'll have. So I will say this, make sure you look back at E, because that's not what would be classified as a polar amino acid. So E would be acidic. So it's going to have a charge on it. So you want to make sure that you look at that.
Oh, yeah.
Yep.
Okay, cool.
But yeah, you're headed in the right direction on this. And so I'll leave you to it unless you have some other questions in here.
I think that's all I had.
Okay. Awesome. Okay, well, we'll see you. We only got 2 minutes left, so we'll see you back in the big room here in a little bit, okay?
Thank you.
Yep. Hey, Jessica, do you have a question?
No, no one responded in my breakout room, so I just left. But to go about this, do you start out by drawing the kind of what we did on the levels of protein structure and the secondary structure, like all the circles, like in like a square type thing, and like I don't know if that makes sense.
It does. I think if I know what you're talking about, yes. And so everyone's coming actually back in the room. So let me go ahead and talk about that. But that's exactly what you'd want to do is kind of like, how can I visualize this? And I think that what you're trying to describe there is how I'm going to show you how to visualize this. So, okay.
Cool, thanks.
Yeah. Okay, everyone should be back now from the rooms. So, and hopefully you were able to kind of at least get a start on this and have some familiarity with this based off of some of the things that we talked about in class, okay? So, when we think about this, let me just go ahead and ask you this, right? So our big question is going to be whether this peptide would form an alpha helix, okay? So let's just, though, first of all, think about this. What are the primary stabilizing forces for an alpha helix, okay? Or secondary structures altogether, right? Just go a little bit more general. What are the forces that stabilize those? Anyone want to venture a guess? You can either speak up in the mic or you can go ahead and put it in the chat. What do you think? Okay, so got some people filling out some stuff in the chat. Let me check that out. Hydrogen bonds, exactly right. Okay, and so these hydrogen bonds that stabilize secondary structures, are they between side chains or are they in the backbone when we think about these hydrogen bonds? Where are they located? What do you think? Give people a little bit of time to answer this. Side chains, okay. Right, let me put it this way. So there's a lot of people actually saying side chains. Let me put it this way. The primary stabilizing forces, right? Those would actually be, and I will say this, there will be side chain interactions, and that's certainly what we want to look at. But when we think about a secondary structure, okay, just like a beta sheet or an alpha helix, okay, the primary stabilizing factors are going to be backbone hydrogen bonds, okay, and by backbone we mean, and the hydrogen bonds that form between those, we mean the hydrogen bonds between the alpha amino group and an alpha carboxyl group, okay. Right, and one of the reasons that secondary structures are so capable of forming and they're so common, in particular in alpha helix, is because they can maximize the number of backbone hydrogen bonds we see, or the hydrogen bonds found within the backbone. Every peptide bond has a hydrogen bond donor and a hydrogen bond acceptor that can be, in an alpha helix, actually satisfied. That hydrogen bonding potential of the acceptor and donor can be satisfied in an alpha helix, okay? So when we look at this, let me think about it this way, and this is why I was asking you those questions, okay? When we look at this, the real question is, well, why don't then all proteins just form one big alpha helix? Okay, so in other words, why are there other types of structures in a protein if the alpha helix is so good at forming these backbone hydrogen bonds? Okay, and that's where we need to step into an understanding of, okay, well, what kind of side chain interactions we have. Now, when I asked you this question, will this form an alpha helix? Really, the question that you're asking is, okay, how are the side chains going to interact with each other? Are the side chains going to have favorable interactions with one another? Or are they going to have unfavorable interactions with one another? Okay, and then that will kind of dictate to us whether we will say that and be willing to say that this this peptide forms an alpha helix. Okay, now how do we visualize this peptide in an alpha helix? That's really what we need to think about, because then if we can visualize it, then we can have some way of saying what side chains will interact and whether they're going to be favorable or unfavorable, okay? So let me do this. Let me go ahead and draw out what would be considered a helical wheel diagram, okay? Now, I haven't used that term in the ACLA 360 recordings or even in the book, okay, but I have shown helical wheel diagrams, okay? And there are two features to a helical wheel diagram. First feature is this: we represent each amino acid in the peptide as a sphere at the alpha carbon. Okay, so we only draw the alpha carbon and we only draw it as a sphere. That's the first thing that we do. The second thing we do with a helical wheel diagram is we grab the alpha helix and we look through it like we look through a telescope. Okay, so let me show you what I mean by this, okay? And let's just draw out here the spheres for these first 4 amino acids, okay? And I'm gonna use blue spheres here. And I'm gonna place them, the alpha carbons of those first 4 amino acids, in a way that we would see them in the helix, okay? So this would be residue number 1, residue number 2, 3, and 4, right? And then we have up here 1, 2, 3, 4, okay? Now, what's hard to see in this diagram is that there's 3-dimensionality to this, right? So as we move from the alpha carbon 1 to 2 to 3 to 4, you're actually moving into the screen, okay? It's moving away from you. And so the sphere 4 will be further away from you than sphere 1, okay? Because it is an alpha helix, okay? Now, the nice thing about this is that because these spheres represent the alpha carbon, we can also then accurately predict where our side chains are gonna be. They're coming right off of this, okay? Now, let's go ahead and draw the rest of this. Let's do the next 3. I'll do a different color here. So this would be 5, 6, and 7. Let's go ahead and put 5 somewhere around here. And we'll put, you know, number 6 somewhere about right here and number 7 somewhere around here. Okay, again, getting further away from us as we draw these. And then our final thing here would be to represent number 8 and 9. Right, and number 8 is going to probably be eclipsed by number 1. That'll be 8, and then 9 is going to be somewhere off over here, almost eclipsed by 2. Right now, we have the only thing we have left to do is just connect these. Okay, and we're leaving out lots of detail here when we make this connection. We're leaving out all the other atoms of the backbone. Okay, but this is a decent representation of the structural features of an alpha helix. Okay, and then we have finally here our last connection, 7 to 8, and then finally 8 to 9. Okay, and that would be a helical wheel diagram of an alpha helix. And from this, we can start to think to ourselves where the side chains are going to be. And then we can answer questions like, will this peptide adopt an alpha helix? Okay. Now, my drawing is super awesome. I mean, let's be honest, right? I'm super proud of myself. I'm going to give myself the old pat on the back. Nice job, Aaron. But I will say this, it's still hard to kind of visualize what the alpha helix looks like, especially when you look at something like this, right? So this is a much better representation of an alpha helix, and you have a very hard time drawing this. But in this case, I'm using essentially the same colors to represent a helical wheel diagram, okay? So we have here, right, that would be our first alpha carbon, and then we can just move all the way down until finally you get to that last pink one down there, and that's the 9th one. Now, here's what's really cool about this, right, is I can take that and go like this, boom, right? And now you see, oh, I can see how it's moving away from me, and I can see the helical pattern of it, right? And all sorts of things. In fact, if I were to do this, let me go ahead and put a sphere around that last alpha carbon, and you can see now there's this line drawn between the first and the last alpha carbon, okay? Now that last alpha carbon, 9 residues away from essentially that first one, it's almost, right, or a little bit more than actually 2 turns away in the helix. Now remember, the pitch of the helix is 5.4 angstroms per turn, right? So if you're exactly 2 turns away, that would be 10.8 angstroms separating the two. Okay, well, you see here this dotted line connecting them, and you see this number 11 in between them. That number 11 represents the number of angstroms separating the 2 atoms that I've highlighted here, okay? All right, so this gives you, hopefully, you know, as I kind of move this around, gives you some sense of what that helix, okay, looks like. And so now we need to ask the question here, All right, what side chains are interacting with each other, and are they having favorable or unfavorable interactions? And then we can answer the question, will that helix form? All right, so let's do this. Let me go ahead and just pick one of these, because there's lots of side chains here and lots of side chain interactions, but let's just pick one, number 2. Okay, now just to give you some idea, I'll draw an arrow, and that arrow represents our side chain coming off of the alpha carbon. Okay, and number 2 is arginine, and it's got a pK of 12.48. And you all think to yourself, that's right, arginine, basic side chain at a pH of 7, right? It's going to be protonated side chain because pKr is 12.48, which means that it has a positive charge. Right? Protonated basic side chains will have positive charge. So we can just put here positive. Okay. You're doing great. I can tell all of you are thinking that, you know, all of you went through that in your mind. Okay. Now, let's ask this question. Okay. What side chains or what amino acids in the helix will it interact with? Is it going to interact with 5? Is it going to interact with 6? Is it going to interact with 1? Is it going to interact with 3, 4, 8, 9, 7, 6? Which ones? Okay. What do you think? So go ahead and give me a possible answer in the chat. What amino acids will number 2 interact with in our helix? Okay, we got some people filling up the chat. Let's look at some of these. Okay, 9 and 6. A lot of people are saying 9. Some of us 5 and 6. 5, right? Very good. Lots of people said 9, and then 5 and 6 and 9. Okay, great. Right. Now let's do this. Okay, so great group of answers, kind of right around what I would expect you to answer. Okay, let's first of all deal with 9. Because there was a lot of people that put 9. And the best way to show this is this, right? So let's look here. This is our second alpha carbon. Okay, that's the one we're looking at. It's arginine. And number 9 is all the way down there below it. Now, that looks like in the helical wheel diagram that those two are definitely going to interact. I mean, they're eclipsed, right? The problem with that is this. They're too far apart. Okay. So when we turn it this way, oops, there we go. When we turn it this way, those are 9.4 angstroms apart from each other. And that's going to really prevent any meaningful interaction that these two would have. That's a long distance in a molecule for things to interact, okay? So I would say this about number 9 and 2, they don't really have an interaction, okay, with each other. Now, however, I will say this, and there was a lot of people that said this, 5, okay, number 5 is right here, and number 2, I think would interact. Definitely. They're only 4.4, they're only 4.7 angstroms from each other, about 1 turn away from each other. They're right on top of each other. Okay, and I would also say this, that it will interact with 6 as well. Okay, a little bit further from 6, but it interact with 6. Okay, now let's do this then. So come back here, and we can start to look at this. I would say that 2 is going to interact with 5 for sure. And I'd say that it interacts with 6. Okay, in fact, the general rule of thumb is that side chains in an alpha helix will interact with other side chains that are 3 to 4 away from them. Okay, whether that be above them or below them. Now 5 will interact with 8 and 9, okay, and it will interact with 1 and 2, right? But let's focus on just 2 here, okay, number 2, and let's look at specifically side chain number 5. Okay, side chain number 5 is a glutamate, and you all say to yourself, ah, yes, glutamate. Glutamate is acidic side chain, pKr 4.25 at a pH of 7. It will be predominantly deprotonated. And acidic side chains, when they're deprotonated, have a negative charge. Nice job, everyone. You're doing great on this. Okay, so I'm going to go ahead and do that. I'm going to put a little side chain here. I'm going to say that's a negative charge. All right, well, we have this interaction, okay, between this arginine side chain and this glutamate side chain that can actually be a positive type of interaction, okay, or favorable type of interaction. It will be an ionic bond, positive and negative, okay? Now, when you look at 6 is alanine. It's really short. It's just a methyl group, okay? And the interaction that alanine has, because it's such a small amino acid with arginine, would probably be negligible in this case. It would interact with kind of the methylene groups on arginine. Arginine is really long, And so any interaction that it has would be either neutral or somewhat favorable, okay, if you look at that, right? But this ionic bond definitely makes me think that, yes, this is an alpha helix that will form, okay? Now, I would have to think about the other side chain interaction, but just based off of this alone, I think is reasonable to think that this will form an alpha helix, okay? Now, just so we know, we've got that one down here, right? There's an arginine all the way down here on number 9, and it's also positive, right? But again, it's too far away to have a repulsive effect on number 2, okay? So given these things, I think it's reasonable to believe that this is going to adopt an alpha helix. Now, just a real quick, look at all these other amino acids, number 1, number 3, number 4, number 6, number 7, number 8, right? So I'll put black stars by those: 1, 3, 7, 6, 4, and 8. All of those are nonpolar, and those nonpolar interactions would be favorable as well, right? All of those are going to have some interactions, like 3 will interact favorably with 6 and 7. 4 will interact favorably with 7 and 8, right? And we could go through this, right? But there's good reason to believe that this alpha helix will form, okay? Right. Questions on that? Okay. If you're still typing, go ahead and keep typing. I'm going to go ahead and move on a little bit, okay? And if I see a question come up in the chat, certainly I'll address it, okay? Now, the next question on the worksheet is, well, is this an amphipathic helix? And an amphipathic molecule, remember, is one that has a domains of 2 different natures, right? So like a fatty acid has a hydrophobic tail and a polar head group. Right, so does this helix have a polar region and a non-polar region? And the answer to this is yes, it does. Right, so if I were to draw a black line right there, you can see that this region up here would be really polar. And this region over here, where I put all those stars, those are non-polar side chains. Right, now some of you are thinking, oh, that's not a polar classification. Those are acidic and basic. You're right, but they have a polar component, right? They're positive and they have a negative charge as well. Okay, so they are polar in that sense, or hydrophilic. Okay, so there's a hydrophilic portion and a hydrophobic portion. Okay, now the importance of that, hopefully you've seen the importance of the amphipathic helices as we start to talk about tertiary structure. Okay, so this would be an example of how this could form. Okay, right, and then finally it says, okay, so if we were to draw the 5 singles of each alpha carbon on this alpha helix, and we would try and place them on a Ramachandran plot, where would we find them? And it turns out, when we go to this, turns out that alpha helices, when they form, the alpha carbons will all adopt the same phi and psi angle to get that helical pattern, and those are going to be sitting right about here. Okay, all right. Now, in the interest of time, let me go ahead and open up the breakout rooms. I'm only going to give you 5 minutes to do this next part. Okay, well, I'll give you 6 minutes. Okay, we'll give you 6 minutes to do this next part. And then we'll all come back together. You're going to essentially use the same diagram to answer that question, but we're going to change pH and we're going to do some mutation. Okay, but before I do that, let me look at the chat. There's a question that was asked. Is this exercise something we'll have to replicate on an exam? When you think about it, to a certain extent, I would say yes, right? It would be looking for you to be able to have some understanding of an alpha helix and the types of side chains that would interact with one another, okay? I would try and make it somewhat unambiguous in terms of the interactions, right? Not something ambiguous though, so, okay? All right, now, let me go ahead and open up the breakout rooms again here. Okay, again, I'm gonna give you 6 minutes. So let me get the time set and then they'll open up. Okay. All right, there we go. And so the rooms are open. So go ahead and jump in those rooms. What's going to happen to this helix at different pH? What's going to happen if we mutate it? Okay, so go ahead and answer those questions. And then we'll come back together as a class. So Grant, right when we come back all together, feel free to take off if you want to get ready for that exam. Just kind of get yourself. Is it? I'm assuming it's virtual.
Yeah, it's like a campus quiz.
Okay. Yeah. So just go ahead and jump off and go to that exam. Help me with the rooms, but beyond that, I don't think you need to hang out. So.
Awesome.
All right. And good luck. Well, stay warm this weekend.
Yeah, it's been ridiculous. I walked home this morning from my girlfriend's place. I did not bring my jacket. It was a bad idea.
Oh gosh. Yeah, you must have been frozen by the time you. Yeah, it was.
Crazy.
Yeah, it was what? It was, I think when I woke up, it was -2 out. So it's a whopping 3 degrees right now, I think. So,
Yeah.
And it's supposed to get colder. Sunday.
I don't know what's happening.
Yeah, it's
Like,
they say there's a mass of Arctic air that has hit the United States. So I guess
it's cold everywhere.
Yeah, it's supposed to be snowing in Texas.
Yeah, I've seen a whole bunch of stuff about that.
Yeah, so
Crazy.
Yeah, it is crazy. All right, how much time do we have here? Let's look at our rooms.
4 minutes left.
Yeah, 4 minutes left. So let's go ahead and start to jump into some of these rooms, see where people are.
Yeah, I'm trying to figure that out now.
We get a question, Mr. Solers. How did you, on the third part of activity 1, how'd you determine where on the graph that goes? Because I know it's like pi and psi angles, but I don't really get it.
Yeah, it's phi and psi angles. So you'd have to look at the exact phi and psi angles of that are reported for an alpha helix. Okay, and I can't remember exactly what they are. I mean, they're definitely in the right region they're in that region that I put them. I can't remember the exact numbers right off the top of my head, but you would just have to look those up, those numbers, and then you would be able to just plot that out, right? You just look at the psi and the phi, and draw essentially a line from the psi angle that would go, it would go, I guess, with the x-axis, right, parallel to the x-axis, and then you draw the phi angle parallel to the y-axis, and where they intersect, that's where there's where you put your dot. So does that make sense?
Yeah, thanks. So if we were asked on a test, we would get the phi and psi angles.
Yeah, I mean, I can't even remember my name, right? So I here's what I would expect, and here's what I want from you, right? That you would know that they're different than like a beta sheet, right? That generally speaking, when we start to think about phi and psi angles, we expect them to be different for the different types of structures because they dictate the backbone structure. Okay, so to get that helical pattern, we have to have a repeating phi and psi angle at each alpha carbon. Otherwise, you wouldn't get that helical pattern. Same thing with a beta sheet. You would have to have the repeating pattern in the phi and psi angles to get the what we call pleated strand. So, so that's kind of the main point of that. Maybe I'll bring that up in the big group. So, okay.
Well, thank you.
Yeah, no problem.
Thank you.
Yeah. Okay, well, I'll go ahead to the next room, unless there's other questions here.
I think we're good.
Okay, great.
One was mutated to E.
Which
So
is glutamate again.
agree. Yeah. B negative. I think it then 2 would interact
Okay, everyone. So welcome back to the main room. So hopefully you're able to get kind of through those questions and make some heads or tails of them. So just real quick, because it's asking you whether TM will change, is going to go up, is going to go down. And just a real quick notation about TM. TM is simply going to be a measure of the stability of a protein. Okay, so let me go ahead and look at this here. There's this graph that we have in our worksheet. And what this is essentially is this, right? You're graphing on your x-axis temperature, all right? But then on our y-axis, percent of maximum signal, maybe not great, okay? But let's do this. This might make more sense. Percent folded, or percent, how would we say this? Unfolded, okay? And proteins happen to undergo an unfolding process, meaning they lose structure as temperature goes up, okay? And so we have here then 100% of the proteins unfolded, and then 0% is of course down here unfolded. Well, the TM is simply this. It's the point where we're at 50% unfolded and unfolded, okay? So higher TMs mean that it takes a larger or a greater temperature to unfold the protein, whereas lower TMs mean that it takes a lesser temperature to unfold the proteins. Those lower TMs then indicate proteins that are less stable, okay? So that was kind of, how we measure stability is by the TM, the magnitude of the TM. Okay, and the first question of this says, what would you hypothesize happens to the TM of this peptide at a pH of 3.5, right? Meaning, what's going to happen to the stability of this protein at a pH of 3.5? Well, pH changes, okay? And when pH changes here, we drop in pH and we actually drop below the pKr of glutamate. And you all say, oh yeah, if we drop below the pKr of glutamate, that means that glutamate then goes from predominantly deprotonated at a pH of 7 to predominantly protonated, which means that it's no longer negatively charged, it is no charge. So we would, in that case, right, lose this negative charge on the glutamate. It would go from negative to neutral, which means that we would lose the ionic bond that it had with arginine number 2. Okay. Right now, that's going to destabilize the protein. We've lost an ionic bond. And that means that our TM then is going to go down in the downward direction. This will become a less stable protein. Okay. All because we change pHs, and pHs, extreme pHs, quite honestly, will unfold proteins. In fact, biologically relevant example of this, when you eat a protein-rich meal, goes right into your stomach, which has a pH of 2. That's an extreme pH. That will denature or unfold proteins. So then enzymes can access them, break the peptide bonds that hold the amino acids together, and then individual amino acids can be absorbed in our intestine. Okay, right. So extreme pHs do that. Right now, the next question says, okay, go back up to pH of 7, right? But this time, we're going to take this positive charge here, 2, and we're going to make that, instead of being an arginine, we're going to make that a glutamate at a pH of 7, which means that this no longer is positive, it's now negative. Well, that's going to lead to repulsion between 2 and 5. And when we have a repulsion between 2 and 5, then TM is going to drop. In fact, this might be so great so as to completely unfold the peptide, okay? Right, so mutation, obviously, if you think about it from this standpoint, genetic mutation will affect function of proteins. A lot of times it will affect function of proteins by disrupting their structure, okay? And that can lead, of course, to quite terrible diseases in many cases, okay? Right, of course, this is not related to that, a disease, but just a mutation, so, okay? All right, so in both cases, I would expect TM to drop, okay? 1, because of repulsive effects, or 2, because we lost the favorable interaction ionic bond, okay? Now, we've only got 4 minutes left, and we got to deal with this fun one, right? I mean, the fun one is super awesome, right? The question is, well, why do you get a temperature when you get sick, right? Some infecting agent like coronavirus or some bacterial infection right, is in you. And one of your body's responses is to actually raise your core body temperature. You get what would be considered a fever. You go from 37 degree, right, normal temperature to, you know, something a little bit higher, 38 or something to that effect. Okay. So, you know, what is that going to do? And that was a Celsius scale, by the way. So 98.6 to, like, say 102 or something like that. Right. So why does your body do that? Well, the whole idea comes back to this graph. Okay. Turns out that proteins in viruses and bacteria, okay, that infect our body, typically will have TMs that hover just above body temperature. So that if you raise your body temperature just a little bit, then those proteins found in the virus or found in the bacteria will start to hopefully unfold. They will lose their function, and therefore the infecting agent's life cycle will come to a halt, or it will slow down, so that then other aspects of the immune system are capable of taking over and removing the infection. That will be the hope of getting a fever, right? Unfold proteins, right? So for instance, you know, of course, bacteria or viruses have to replicate their genome. Well, if they have a protein encoded in their genome that does that, replicates their genome, that gets unfolded in response to the fever, then that genome can't be replicated, and they can't then continue to pass on, you know, their genetic material to the next generation, and therefore the life cycle stops. Like, that's the whole idea, at least, that we would think about with the fever. Now, some of you are sitting there saying, "Oh yeah, but that happen to the proteins in our body too if we raise our temperature?" Yeah, it does. Unfortunately, many of our proteins are unstable at higher temperatures, even slightly higher temperatures. However, in our cells, there's a whole group of these proteins called heat shock proteins. And these are proteins that were discovered that when a cell undergoes increased temperature, there are genes that will be expressed. And those heat shock proteins actually stabilize other proteins in the body, stabilize or refold them. Right? They won't do that for an infecting agent, but they'll do it for our proteins. Okay, so there's a protective mechanism in our own body that found in these heat shock proteins. Of course, you know, extreme temperatures, extreme fevers can be extraordinarily dangerous, and so you want to seek out medical help in those cases. So, okay, all right, so super awesome stuff. Like I say, right? Especially when you think about this on a cold day, right? What else would you want to be doing besides in here talking about biochemistry and, you know, TMs and wonderful stuff like that? Okay, now here, let me do this. Okay, last thing to do for the class today. I'm going to unlock this question. You know, what do you think? What are some Q&A stuff that you've got? Go ahead and put some questions on here. All right. Again, I'm going to hold a special Q&A session on Tuesday at noon. If you have to make it a little bit late, no big deal, that's fine. But we'll be hopefully addressing a lot of these questions. So, okay. If you don't have anything, you don't have any other questions, by all means, feel free to take off. Have a great weekend. Stay warm, and we'll see you next week. Okay. Take care, everyone. And if you have questions right now and you want to just kind of look around, I'll hang out in the Zoom meet for.
I think I have a quick question on TM, just for clarification.
Yeah.
So exactly, the TM is just at what, like the threshold of what temperature is going to cause a protein to denature?
Yeah, I mean, we're always picking these points where it's 50% done. Okay. So
right.
The TM is where you're at 50% unfolded. Okay. That's
oh, I see. Okay.
Yeah. So we would say, if you know, if you have 100 protein molecules in your vessel, right, your reaction vessel, 50 of them at that temperature are unfolded at what it's called the melting temperature with the TM. Okay.
Oh, okay.
You know,
that makes more sense.
Yeah, great. And then if you look at the shape of the curve, right, just to kind of expand on that a little bit, you look at the curve, it's called a cooperative process. And there's a really, you know, there's this huge jump Okay.
Mhm.
So just small temperature changes right around that level will lead to either a lot more protein being folded if you're going downward, or a lot more protein being unfolded if you're going in the upward direction.
Oh, okay. That makes a lot more sense when you explain it like that, because now I could see that it's like a really small window that it seems like that it just jumps from like 20 to 90 or 100%.
Yeah, exactly. Right. So then you think about a fever and you're only increasing temperature by 2 or 3 degrees. And like, what is that going to do? Well, if the proteins, right, TM is sitting right around 38 degrees Celsius, right, getting up to 39,
Right.
you're going to go, you're going to go up to like 75% of the protein unfolded. And that will really affect it.
Nice. Okay. So, does every, I guess, protein just kind of have like an associated TM with it then?
Yeah, and it's different for every protein, and it's based off of the types of interactions. Some proteins, like the one that you're looking at in that graph, called ribonuclease, its
Mhm.
TM looks to be like, let me see here, it looks to be hovering around 58 degrees Celsius. Okay,
Right.
some of them are way lower than that. Some of them are way higher than that. You have to look at like boiling temperatures to get them to unfold.
Ah, okay,
And then,
cool. And
yeah, go
oh,
ahead
I'm
again.
sorry, go ahead.
Well, we think about that in terms of stability, right? When we talk about protein stability, the higher
Mhm.
temperatures means that there are lots of interactions that are holding that protein together. So if you think about like organisms that are found in Yellowstone's hot springs, right? Those things thrive
Right.
at like 98 degrees Celsius. Okay, well that means that their proteins, the majority of their proteins, have TMs that are well above boiling temperature. So what's
Oh,
different
wow.
with those proteins than our proteins? Because we would die at that temperature, right?
Yeah.
I mean, most of our proteins would unfold. So there's investigation of like, well, what makes those proteins more stable than others? Okay, and it comes
Nice.
down to like more ionic interactions or greater hydrophobic cores or something to that effect. Yeah.
Right. Okay, cool. That's a really cool context to put that into. That makes a lot more sense.
Okay, great.
Awesome. Thank you.
Yeah, take care.
Have a good weekend.
You too.
Can I bounce off that? I think this might just be more of like a clarification thing, but so then would that mean that like all of the proteins that make up our bodies are like have a TM that can withstand like basically what we would consider a normal fever, and then the reason that like an extreme fever is considered extreme at the temperature that it is because that temperature is above the majority of our proteins TMs. I don't know if that was the best way to word that.
Yeah, no, I understand what you're saying. So the danger comes that the higher the temperature goes, right, the more and more unfolding we have. And we start to then eclipse other proteins TMs as well, right? And then you have a more massive problem on hand. Now, I will be honest too. I mean, there are other physiological processes that are going to be negatively impacted by higher temperatures. Okay,
Sure.
besides just proteins folding and unfolding, although that's a major aspect of this, certainly. Okay, so when you talk about extreme temperatures being dangerous, right? You're thinking about protein unfolding. You're thinking about ionic charge across the membrane that might be disrupted in that environment. You might think about even membrane fluidity, how that might change and how that will change cell permeability. So there's lots of things that can be affected by this. Okay, all of it though related to very similar ideas, right? Biological structures are stable within certain temperature ranges, and as those ranges go up, as that temperature goes up, then you're going to, of course, have significant problems. So.
Okay, that's really cool. And then I also wanted to clarify, I had to hop off for a minute while you were explaining activity number 2. So for the second question about the mutation of the second amino acid to E, was that just like a decreased TM due to now you would have like a negative and a negative interaction on the 5th and 2nd?
That's
okay.
Right. Yep.
Awesome. Cool.
Yep.
Thank you so much.
Yeah, no problem.
Have a good weekend.
All right, you too. Take care.
Thanks.
Yep. You're welcome.